9, so let's get started. I probably shouldn't have told people there won't be clicker questions today. Maybe that's why so many people aren't here. But that means there'll be extra clicker questions on Friday. Yay! Yay! So based on today's lecture, which is being given by someone who knows a heck of a lot more about Flaviviruses than I do, which is why I invited him to come and give this lecture. It's not because I'm lazy and I didn't want to do it. I was hoping that you get a little bit more information this way. Alec Hirsch brought to us at massive expense from the Vaccine and Gene Therapy Institute at OHSU. Alec did his undergraduate work at Swarthmore, then a PhD at Princeton with Tom Schenk, and then came to OHSU with Jay Nelson. Is that the start of the VGTI?
Yeah.
Yeah, pretty much the beginning of the Vaccine and Gene Therapy Institute, which is out at the West Campus of OHSU, where they also have one of the largest non-human primate research centers in the country, where they're doing some really, I think, really fascinating work with all kinds of different viruses. Dr. Hirsch has worked a lot with West Nile virus and dengue, and not surprisingly, the Flavivirus du jour, Zika virus. So I will let him take things away from here. Again, feel free to ask questions as you move along, and I'm sure Alec will be happy to try and answer those. If there's something that I feel he could be potentially doing a little bit more of, I will yell and scream in the back. So that's my role here. So, Alec, all yours.
Okay. Well,
do we need the lights down a little bit, or you think it'll be all right?
okay. I guess yes. Okay, so that's weird. Anyway, Flaviviruses. So this is what I spend a lot of my time thinking about. Please feel free to stop me at any time with questions or comments. So just to give you an overview of the family Flaviviridae, shown here is a map sort of based on virus phylogeny based on sequence. And the Flaviviruses, so they have several things in common, and then there are certain divisions that have some differences. So they're all RNA viruses with a single positive sense RNA. They're all enveloped, and so that applies to everything on this list. So there are different, so that's the family. So then there are different genera within this group. So the genus that's actually keeps the name Flaviviruses are these vector-borne viruses here. It's either mosquito-borne or tick-borne Flaviviruses. I'm going to focus mostly on the mosquito-borne Flaviviruses, and those include things like yellow fever and West Nile and dengue virus and the Zika virus as well. Also within the family are a group called the pestiviruses, which are actually tend to be viruses of livestock and are of a fair amount of importance in agricultural settings. And then finally, I'm going to end the lecture sort of moving away from the mosquito-borne Flaviviruses and talk a little bit about the Hepativiruses. The name of the genus comes from the primary member of that family, Hepatitis C virus, which is, so it has the basic similarity, but the life cycle of HCV and the pathogenesis is quite different from the mosquito viruses. Okay, so that's good for that slide. So we're going to start with yellow fever virus. That's where the family gets its name. The Latin word for yellow is flavus, and it's the prototype of the family. So I just cut out a page from a 1793 description of a yellow fever outbreak in Philadelphia. And if you can read it, notice it has that kind of crazy thing they do where all the S are Fs. I've never understood where this comes from, but and it takes a while to get used to it. But notice that So the author here, Matthew Carey, is describing sort of the typical symptoms of yellow fever virus. Signs of putrescency appeared at the beginning or before the end of the third day. There's often one of the characteristics is what they call black vomit. That sounds pretty cool. It's also described as coffee ground-like, and that's actually from hemorrhaging into the gut. And then the disease, gets its name from the universal yellowness, which comes from disease of the liver, and then the fever and all the prostration of strength, and also the disease course often included the febrile symptoms, then a day or two where they began to wane and then came back even stronger, where you begin to have skin that assumes a tawny color mixed with purple, more of the black vomit, hemorrhaging, hiccups, anxiety, coma, and death. So this was a big deal in colonial America, and actually throughout the New World through the 18th and 19th century. It is believed that yellow fever, it's known now that it's endemic in Africa and probably was brought to the Americas through the transatlantic trade, primarily the slave trade. As I said, the results of infection can result not only in the characteristic yellow color and hemorrhage and vomiting, but also organ failure and death. So for a long time, there was no particular way to treat this. Sort of things that were tried in 18th century Philadelphia included massive bleeding of the patients. And the cause was generally ascribed to things like, bad air and ill humors. You can find a lot of references to this in the writings of Benjamin Rush, the colonial era physician in Philadelphia. And this largely continued till roughly the middle of the 19th century, when people began to associate the disease with the presence of certain mosquitoes. And although there's probably evidence of this hypothesis a little earlier than 1881, probably the most complete laying out of the mosquito hypothesis is a Cuban scientist named Carlos Finlay, who specifically, wrote down in publication the idea that the virus is transferred from mosquitoes to people, and then the mosquitoes pick it up from infected patients, and so you have a cycle of transmission. This was more formally proven, generally Walter Reed, who name now is at one of the primary military hospitals in the DC area. But so he was a doctor working for the U.S. Army, and first he initially, started looking at the causes of yellow fever and examining the possibility that various bacterial agents may be responsible for this. That didn't pan out, and then he was able to show that actually it was a filterable agent, which at that time meant, viral. It can move through the sort of filters they use that would exclude things that are bacteria size or greater. And viruses had recently been described, or slightly earlier in the century, so they had an idea of what this sort of thing was. And I don't know if Ken had talked about this a little earlier in the class in terms of the initial discovery of virus, but I think that preceded Walter Reed, who was working at about 1900 by, 40 years, something like that. Anyway, so they also explored the possibility of mosquito transmission, and they would isolate a single mosquito in a test tube and allow it to feed from an infected individual, and then pass it on to volunteers. Volunteers, yeah. And a lot of this work was done by Dr. Reed's colleague Jesse Lazier, who actually did the experiments showing that you could pass yellow fever in this way to an uninfected individual. One of the seminal discoveries was that you had to wait about 12 days after the mosquito fed before it became infectious. And he was able to pass yellow fever to himself from a mosquito that had fed off an infected person, had incubated 12 days, and then when it bit Lazier, he contracted yellow fever. And then he died. This is when science was much more hardcore than it is today. But anyway, this apparently convinced Walter Reed that the mosquito hypothesis was in fact true. And Reed did a lot of further controlled experiments demonstrating lack of transmission without the mosquito vector, things like where you carefully remove all the mosquitoes from a room, place mosquito netting around it, and then have a volunteer, say, sleep in the bed of a yellow fever patient who had just died from the disease. And they found, without allowing any mosquitoes in there, the patients emerged healthy. So this observation, actually, just knowing that it was that you could control the mosquito vector and prevent the disease, was key to controlling yellow fever, primarily, in the Americas, and also key to things like the building of the Panama Canal, which they had been trying to do, and workers would continually contract yellow fever and become too ill to work or die from it. But the advent of just being able to try to get rid of the mosquitoes, get rid of their breeding grounds, protective clothing and netting and such, did a lot to allow advancement of the Panama Canal and humanity in the New World. Next. No, that's going backwards now. There. Okay, so just to summarize sort of the major findings of Reed, and this is true of other flavivirus as well, or at least those that cycle between humans and mosquitoes, like dengue virus, which I'll talk about in a little bit. The mosquito acquires the virus from somebody who has been infected, or in some cases, if there's an animal host, it acquires it from an infected animal. In the case of yellow fever or dengue virus, probably some sort of primate. There's then the extrinsic incubation period, in which the virus replicates within the mosquito, and then the virus titer within the mosquito is high enough that it then can pass it on when it bites a naive person, who then there's a short incubation period within the person before they become symptomatic. There's a period of viremia in which they're capable of passing the virus on to another mosquito, and generally the illness occurs sort of midway through the viremia period, that's presence of virus in the blood, and beyond, depending on the actual virus or the severity of the disease. So, control of the mosquito was very effective, but even more effective was the advent of the yellow fever vaccine, which was first described by Max Taylor and Smith. And what they did is there was some knowledge that you could, or hypothesis, that you could pass a virus in a cell in which it normally doesn't grow. And this would change the virus, at that time they thought, just in some way, which would render it less dangerous when you infected an individual. So they followed this strategy and passed a pathogenic strain of yellow fever roughly continuously for 3 years, primarily in cells derived from chicken embryos. And one of besides the what they call the viscerotropic, or liver tropism of yellow fever, there are also various problems with disease that could be associated with the virus getting into neurons. So they actually specifically removed the brain and spinal cord from the chicks, ground them up, cultured the cells, and grew the virus, and in that. So that went on for about 3 years, maybe 200 passages, and they found that the virus was attenuated. And they did this test in a monkey, a rhesus macaque, I think, showing that where under normal circumstances the wild-type virus would cause something very akin to yellow fever in the monkey, the passage virus was no longer able to cause disease. But then they thought this could be effective as vaccine, so they began to do these tests and found that it was, in fact, a very effective vaccine. Much later, when people were looking at the mechanism of what's actually different about this virus than the parent, it's still unclear. There's just a very small change in the genome. So the single-stranded RNA is about 10,500 nucleotides long. The attenuated strain has a difference of about 67 nucleotides within the reading frame. That means 32, or that changed about 32 amino acids. So which one of those or what combination of those is still an open question. Another possibility is actually just the loss of diversity, in that if you get infected by a mosquito in the wild, you're actually getting a pretty wide variety of differences in the sequence of the virus. So basically we call it a swarm, whereas the vaccine strains are much more homogeneous. And it has also been proposed that it's this lack of diversity which may contribute to the lack of pathogenicity. But as I said, the vaccine is extremely effective and has done a lot to sort of stem the tide of yellow fever in the world. It is not perfect. There are still some adverse effects, and it is a live virus, and it is not recommended for immune-suppressed individuals, which used to be less of a problem, but now that we have people with HIV who are living longer because their disease is managed, but still may have instances of immune suppression or various transplants, or even just anti-inflammatory drugs that have immune suppression phenotypes. Things like this are things that need to be watched when vaccinating individuals. Okay, good? All right. The virus is not gone. It's still present in the tropical and subtropical world, primarily South America and Africa, and probably still endemic within primate populations in these regions. And I want to do a brief interlude on the mosquito vector of yellow fever and dengue virus. This is the primary vector, Aedes aegypti. It is exquisitely evolved to interact with humans. Humans are its favorite host. Its primary place of residence are little pools of water in and around human habitations. It doesn't move very far. It tends to just stay close in to humans, so it's very prevalent within urban centers. A secondary vector for yellow fever or dengue virus, and maybe some of the other mosquito-borne viruses, is this Aedes albopictus, which also tends to feed in the day. I borrowed this slide from another researcher. When he says it's a Catholic blood feeder, I think he means that it will sort of non-discriminately feed off different, people or animals, sort of all-encompassing. So little c Catholic blood feeder. Just to be clear about that. And you'll find this in sort of more natural settings or in close association with humans. Generally, things like dengue virus track much more closely with the Aedes aegypti than with albopictus or other Aedes genus mosquitoes. So I said that mosquito control had been particularly important in the control of yellow fever and subsequently dengue virus as well. And because of this, and you can see, so this is the distribution of Aedes aegypti in Central and South America and up to North America. You can see that sort of after the 1930s, a lot of programs were begun to try to control the mosquito, which were extremely successful during the 1960s into 1970, as shown in the middle panel in this graph. And by now, most of these programs have collapsed. There are potentially a couple reasons for this. One might be that a lot of this was achieved through really indiscriminate use of DDT, and the programs could be conducted efficiently because a lot of these governments were authoritarian military regimes, so you couldn't really complain when they sprayed DDT at you to control mosquitoes. And a lot of those have gone by the wayside, which is a good thing, except maybe from the point of mosquito control. I won't comment anymore on the benefits of fascism, but so the mosquito is back, and this is a problem, and we can see sort of with the explosive spread of Zika virus, which is also spread by the Aedes aegypti, just how how big a deal this is, and how other maybe less chemically aggressive ways of mosquito control really need to be carried out. Okay, so moving on to the dengue viruses. I think I skipped over an earlier slide which just had the basic outline of the talk, but as you may have gathered, what I'm going to do is probably talk about some of the different flaviviruses and various salient points regarding their disease or biology, and then kind of move into a more molecular aspects of flavivirus biology about the second half of the talk. So anyway, the dengue viruses actually consist of a group of 4 different serotypes, which can really be considered 4 different viruses. And the concept of the serotype is rather complex, but these things are all defined and were defined before there was sequence analysis, all defined by antibody response. And it becomes very difficult because there are antibodies that you can get from infected people that will recognize all of the flaviviruses. In fact, that's initially how the family was described. And then there are antibodies which are specific for certain, what they call serocomplexes, like the dengue viruses. And there are other antibodies that will discriminate between 4 major groups of dengue viruses. And those are called dengue 1, 2, 3, and 4. Is everybody with me? Because at this point I always tend to lose people. But okay, so there are basically 4 flavors of dengue virus that used to be present in somewhat restricted areas, but now with travel and the spread of the mosquito, you probably find in places like Brazil or Southeast Asia that multiple serotypes are circulating at any one time. All of them can cause dengue symptoms, which are, I'll go to it later, but are first are described as dengue fever, sometimes called breakbone fever, which is a severe and debilitating febrile symptom, and then there's more severe types which are involve hemorrhagic symptoms that cause capillary leakage, loss of blood pressure, can lead to shock and even death. Within these different serotypes, you'll have different strains that have genetic variation, and there's the potential for some strains to be more or less pathogenic than others. So when one particular strain goes through a population, the extent of disease may vary depending on multiple factors, but some of them may be regarding the virus itself might be nastier. So the current classification of dengue disease, and you maybe have seen what they call the older classification, is dengue fever and dengue hemorrhagic fever and dengue shock syndrome. Currently, medical doctors are advised to classify it as dengue without warning signs meaning the progression. Are there certain signs that could indicate the progression to hemorrhage? Dengue with warning signs, which would be signs that hemorrhagic symptoms and shock may be on the horizon, or severe dengue when you're already at that point. And one of the reasons for reclassifying the dengue disease severity is basically, in many cases, dengue is a mild fever. It can become a more severe fever that's more self-limited. But when you get to the hemorrhagic symptoms, a patient is not likely to die, but if they can get the proper medical support, which usually means hospitalization. So one of the major problems for doctors in dengue and endemic areas is knowing when somebody comes in with something that looks like dengue fever, who do you send home and who do you check into the hospital, which is why sort of being able to anticipate the disease before it gets that bad, or if it's likely to get that bad, is a big issue in the dengue field. Let's see, so this is the distribution of the thing. It's the dengue viruses are pretty much endemic surrounding, the equator and the tropics of Cancer and Capricorn, so the tropical and just subtropical world. Generally, the statistics I use for background slides or to write in the first sentence of grant applications is 2.5 billion people, or 40% of the world's population, are at risk for dengue infection. That's the percent of the world's population that lives within this area. The number of infections per year is estimated at just under 400 million, of which about a quarter of those are actually symptomatic, so dengue fever or worse. In fact, about 500,000 are severe, requiring hospitalization, and that are potentially fatal if care is not sufficient. There's no, I used to say there is now a potential vaccine, and I'm going to talk about that a little later once we talk about Flavivirus sequence, so just hold off on the dengue virus vaccine. There's no particular antiviral drug or treatment that can be used. Treatment is all basically supportive at this point. This is just a slide I took out of the CDC's weekly publication. This is from 2010, showing autochthonous, that is, local transmission of dengue virus in the Florida Keys. So it is actually present within the United States, and this isn't traveler associated. These are infected mosquitoes within the state of Florida biting people and passing on the disease. And we've seen this occur in various regions of Florida primarily since this time, in and along, not only just on the Keys, but on the Maine Peninsula along the Atlantic coast. Last year there was a pretty big dengue outbreak, so the disease, although it's primarily located, in Central South America, in the Western Hemisphere, it is kind of nipping at the heels of the United States as well. Let's see. Okay, so this, and there's another concept which I want you to stop me if I'm not entirely clear, because this can also get a little bit confusing, but it's generally believed that once you're infected with dengue and it goes away, you're probably immune for a long time, maybe your lifetime, to reinfection with that particular serotype. So if you were infected with dengue 2, you should be immune to dengue 2. And much of this data comes from experiments conducted during World War II by Albert Sabin, who you may know as the oral polio vaccine guy. And he conducted a lot of experiments, again using human volunteers, whereas actually, I guess because it was during the war, they didn't actually use soldiers as volunteers in this case, the way Reed did.
Sabin's volunteer pool were prisoners, psychiatric patients, and medical students.
It's about part of the course.
Yeah, you figure out which one had the least choice in what they were doing. But anyway, so he observed initially following dengue infection that you probably had an antibody response which would recognize all forms of dengue, but that would go away, and then you would be left with something that primarily recognized just that specific serotype, and that's the long-lasting immunity that grants you immunity to just the homologous serotype. And it's also been observed that if you're infected, after having been infected with one type of dengue, if you're infected with another type, the odds of having severe disease are much higher. So most, not all, but most of the severe dengue episodes are secondary infections. That is, infections with, so if you were infected with dengue 2 and then you're infected with dengue 1, 3, or 4, the chances of that disease being worse are much greater. And the mostly agreed upon reason for this is this hypothesis of antibody-dependent enhancement. And what this is, and again, is the idea is if you have an antibody recognizing one serotype of dengue virus, so it will bind to that virus if you're infected and prevent it from infecting cells. In the case of being infected, say, with a different serotype, so if you had been infected with dengue 2 and now you're infected with dengue 1, the antibodies kind of recognize the virus. They will bind to it partially, not enough to actually prevent infection of target cells. And further, because the antibody is bound to the virus, certain cells have receptors that bind to the constant region, the conserved region of antibodies, things like macrophages and dendritic cells. Their normal function is to, say, pull in things like that are antibody-coated, say, like bacteria or other foreign agents. So they'll wind up doing this with the virus, but then the virus is able to infect that cell. So what happens then in the case of antibody-dependent enhancement is the pre-existing antibody response to another serotype of dengue allows for more cells to become infected when you're infected with the second serotype. Yeah? All right.
Alec, just maybe to repeat this, because I think it's kind of a funky concept. It's something I've always had trouble following. And the idea here is that if you have complete antibody reaction, then you have a different process than if you just have the macrophages and dendritic cells picking them up. Because if you've got complete antibody coverage that aren't macrophages and dendritic cells also picking up these viruses, so what's the difference there?
The difference is probably, so those antibodies are able to neutralize. So and they're probably binding in ways that are actually preventing any subsequent membrane fusion of the protein, of the envelope protein with the cell membranes, and I'll get into this in a little bit. So that virus is covered to the extent where it's been rendered inert. And so certain antibodies, again, what we're dealing with here in the human is a polyclonal response with lots of different antibodies, but it's clear that there are certain antibodies which are much better neutralizers than others for certain serotypes. So,
the basic difference between neutralizing antibodies and just an antibody response.
And just one that binds and
then leads to
does not neutralize. So this can be modeled actually in mice, and that's the data slide I included here. And this is data from Sujan Shrestha, who's at the La Jolla Institute of Immunology down in San Diego. But what she had done is first they had developed this model of dengue infection in mice, and it's an adapted virus and it's adapted mice, but I won't go into that. But basically, if you take this particular dengue 2 strain and infect these mice, so this is just shown as survival. So here, at day 0, 100% of your mice are alive, and then by day, 10, 11, all of them have died from dengue 2. So that's the same data in each one of these graphs. So yeah, this strain is dengue 2. So if you at the time of infection, also give the mice immune serum from mice that had been infected with dengue 2. In this case, it's not 100% protective, but about half of your mice are protected, right? If you give them immune serum from mice that had been infected with dengue 1 or dengue 3 or dengue 4, that's these lines here. So then the mice die much more rapidly. And she goes on to show that you have greater viral load in certain tissues, and different groups have sort of tried to target sort of different cells that are actually making the different, that are actually the ones that are sort of giving you that extra boost of virus that yields to greater lethality. But this is the concept sort of modeled in these mice, and this is highly reproducible, and you can do it even with monoclonal antibodies that neutralize, or just if you give not enough antibody, it makes things worse instead of better. Yeah.
This might be out of your wheelhouse, but I seem to recall something, and the guys trying to work out the 1918 flu mortality, that there was a similar thing, but they don't have the first flu.
We'll talk more about that when we talk about influenza.
Yeah. Okay. Yeah. That is out of my wheelhouse. I'm sorry. So we'll just go back. Yeah.
Why it's really difficult to develop a vaccine for this virus?
That is exactly right. In fact, I'd written that on my previous slide on the bottom. That's why it's causing problems for vaccination. Is that when you vaccinate against dengue, you have to get all four, because if not, you're potentiating greater disease for whatever serotype you did not effectively cover. Okay, all right, so dengue virus, we're going to do a little West Nile, Zika, molecular, and then HCV at the end. So I just want to give this example of the kind of cool thing about West Nile is just the way it spread. It showed up somehow in New York. It's still unclear to me, and I think it's unclear if what was transported was a mosquito that had been infected with West Nile, or if it came in some other host, which was then bitten by a mosquito. But sometime in the summer of 1999, a bunch of birds at the Bronx Zoo got sick and died of some sort of neurological infection, which was later identified as West Nile, a virus that had been discovered some 60 years previously, and generally had not been thought to be too dangerous, although recently it had been circulating in the Middle East in a more virulent form, and that's probably what showed up in the U.S. So after that, sort of a pretty cool door-to-door study by the New York Public Health Department, sort of just asking people mostly in and around the Bronx and Queens, where a few people had shown up with this neurological disease that had been identified as West Nile. They went, did blood draws to figure out who had West Nile antibodies, asked who felt sick at some point, and they figured out probably during this first summer, maybe about 250 people had been infected with West Nile virus. But the virus stayed and it spread. So you can see where this was by 2001, it had gotten about as far as the Mississippi. And then by 2003, it had finally kind of crossed throughout the entire continental United States. This is from 2014. This is a slightly different map. So this is just showing human diseases that are characterized by severe West Nile, which is when it gets into the brain and you have neuroinvasive disease. So unlike the other, or yellow fever and dengue, this virus is not evolved to grow in humans. It is primarily a virus of birds, and certain birds are the usual host. And it is actually transmitted by a different mosquito. These are Culex genus mosquitoes, which are a little less likely to bite humans than the Aedes mosquito. They're also a little bit more wild and found less frequently near human habitation. Nevertheless, when one of these mosquitoes does choose to feed on a human, there potentially a few things can happen. In most cases, the disease is asymptomatic, so you don't even notice. In some cases, there is a generalized flu-like illness, which passes on. But in the most severe cases, the virus is able to cross into the brain, which causes all sorts of problems. So that's the 1 to 2,000 cases of neuroinvasive disease per year. And although probably only 10% of these cases are fatal, I mean, that's still bad enough, but the risk of long-term consequences from this type of encephalitis is pretty high. So, so any sort of incidence of neuroinvasive West Nile disease is something to be concerned about. Also shown here is it was also recognized that similar things happen in horses for some reason. So these horses are also subject to West Nile disease. They're also accidental hosts. The reason I chose to have this picture of Abe Vigoda here is that the disease is, well, once somebody dared me to put a picture of Abe Vigoda in a slide, but the more important thing is that the elderly are the most susceptible to West Nile disease because of certain things that, changes in the immune system with age that makes them less able to deal with this particular virus. Good. All right. So this is moving along slower than I thought. I better speed things up. Anyway, so quickly, Zika virus, which is in the news all the time. It was discovered, so in 1947 was the discovery of Zika virus. And this was done by a team of American and British scientists who were working in a research station in Uganda. They were actually looking at yellow fever transmission, so they took rhesus macaques, which are actually not native to this region, put them in towers like this up on the top, and then every day they would climb up the tower and see how the monkey was doing. So one of them was found in 1947 to have spiked a slight fever, so the monkey was taken down off the thing and off the tower and brought to the lab. And they were able to isolate a virus from this animal, and that is the Zika virus, named after the Zika forest, which was the region where that tower was. I should just point out that if you look at some of these early publications, they have this map of the region of Uganda that they use for their virus hunting. And like every region of this thing is a virus. So there's the West Nile region up in the one part of the map. There's some Licky Forest, the Rift Valley. Everything of these has contributed some sort of virus to, for the rest of us to work on later on. So they found this thing, and they were able to show then by mostly using antibody-mediated experiments that it was different from dengue virus, which they're already aware of. And they're able to show that a large portion of people in East and also in West Africa were seropositive for the virus. So it did circulate in the human population, but incidences of Zika virus disease were very rare. The monkey who was up on the tower, he had a fever for 2 or 3 days, and then he was just fine after that. They passed the virus on to another monkey, which did develop an antibody response, but that monkey never got sick. One of the sort of clear descriptions of a human disease involve a researcher who was actually working in this area, trapping mosquitoes, looking for various, new viruses, and he fell ill with a fever-like symptom, which he was able to attribute to Zika virus, again, based on studies of his antibody response. So that guy got to at least publish the paper describing his own disease. But again, these things were sporadic. They're generally mild, self-limited. It was found that later on it had been described in Asia, or found in Asia, but again, these were a few cases of disease that looked like a mild dengue. And so nobody had heard of Zika virus until now. Anyway, it had been discussed roughly, so this is how things stood until 2007, when there was a large outbreak of febrile illness on the Micronesian island of Yap, which is there, that people found after eventually ruling out dengue virus that it was, in fact, Zika virus. So some people fell ill here, but everybody got better. And then further spread, probably both these were independent spreads from Southeast Asia. Then in 2013, it showed up in Tahiti and French Polynesia, where there was a larger population and a large number of people who fell ill with a febrile type illness identified as Zika. And this also was associated with a fair amount of what's called Guillain-Barré syndrome, which is a neuromuscular disease characterized by paralysis, in some cases the need for mechanical ventilation, that was associated with Zika virus symptoms. And actually, there were also, this wasn't noticed until later when people went back and looked, but this outbreak was also associated with a certain number of birth defects. So then in 2015, we come to the large outbreak in Brazil, which was possibly acquired by a traveler from French Polynesia. It's been suggested that it had something to do with a kayaking team which came to some competition in Brazil. I'm not really sure how good the evidence for that is. But so, as you know, the incidence of Zika virus in Brazil has gone through the roof. If I go back here, yeah, okay. So based on it's not actually a reportable disease, so we don't have really good numbers, but the World Health Organization estimates that maybe 3 to 4 million people have been or will be infected during this current outbreak. It's associated with this paralysis syndrome, that Guillain-Barré syndrome, which I described. And thus far, there are about over 7,000 cases of microcephaly, or shown here, which is this characteristic sort of loss or failure to develop of certain parts of the brain, and this sort of small head or microcephalic phenotype. This, so far as we know, it's the association is now quite clear. It's not clear, at least to me or what I've read in the published data, exactly if it's associated with infection at any one particular point of the pregnancy. There are other neurological defects that have also been described in fetuses, and in some cases, the severity of the microcephaly is probably goes all the way from infants that just wind up stillborn to ones that are alive and probably will continue to live, but will have to deal with various symptoms then for the rest of their lives. So this is becoming a major public health concern. Yeah.
When you say it's not reportable disease, what does that mean?
So certain things, the various public health departments require that doctors who observe a confirmed case of the disease then notify the local or federal public health department, so they have good records. This was not initially the case for Zika virus, so a lot of this happened anecdotally before people were really able to get a handle on exactly how much. So this number of 3 to 4 million people is very much an estimate.
Is it now? Is it now?
I don't know what the current status in the, at least in Brazil or the other Central American countries. They are certainly trying to keep a greater track of it. The CDC has a presence in Puerto Rico where the disease has appeared, and I believe they're trying to keep careful records of, say, the number of cases and follow all cases of pregnancy in that case. So it depends on the country to some extent. Yeah.
Do they know what caused the increase of pathogenesis in this mutation?
A good question. And let's go to the next slide. No. The answer is no. That's why I put the question up here. We don't know what caused the increased pathogenesis. Is it a genetic alteration of the virus, increasing virulence? It is. So this virus had not been present in the Americas before. Is it just introducing a new virus into a population where you have rampant mosquito presence and a completely naive population? So every person the mosquito bites would be able to then become viremic because there's no pre-existing immunity. Is it co-circulation with other viruses? Dengue virus and the alpha virus chikungunya are also endemic in these regions. And of course, the connection with microcephaly is pretty well accepted at this point, but the cause is still an unknown. Okay, so I left myself about 15 minutes here. So this is the Flavivirus life cycle, right? So all of these things.
Worst I can cover some of this too.
Okay. All of the viruses I talked about do the same sort of thing. They get in, finding some sort of receptor that's generally unknown. When you're an enveloped virus and you get into the cell by a receptor, you are within the cell, but now you're trapped in some sort of endosome, so you have to then fuse with the membrane to release the nucleocapsid. This all happens in the cytoplasm. There's not really any important nuclear association, although some flavivirus proteins do show up in the nucleus. We don't know why. But anyway, so that single positive sense RNA is released into the cytoplasm. It's positive sense, so it can function as a messenger RNA. So it's translated, and you make a single polyprotein, which is cleaved into various functional subunits, including the replication complex. So then some of these proteins are able to go back to these genomes and copy them into a negative strand, so a complement to this one. And then this complementing negative strand is used by the replication complex to make more positive strands, which then associate with the capsid protein, acquire an envelope by budding into the ER, and then it moves through the secretory system. Eventually it gets to the trans-Golgi network where it sees the protease furin, which is required to cleave a viral membrane, the viral M protein. That's a final maturation step, and then it gets out. This is the structure of the flavivirus. It is enveloped, shown here in its fully mature form. You have the envelope protein, or E. Each one of these colors is a dimer, so two of them lying next to each other. It's generally described as T equals 3 symmetry. But this is the envelope. It's not the capsid. The nucleocapsid is actually, as far as anyone knows, fairly unstructured. It's just a protein and RNA kind of bound up inside. So this symmetry comes from the arrangement of the envelope proteins lying on the surface of the cell. This is another look at the envelope protein. Again, this is a dimer, here's one and then another one lying next to it, with probably this hinge region being important for receptor binding. So flavivirus entry, I copied this slide from somewhere. This is the whole life cycle, but I'm just going to focus on this entry part here. So, as I said, we start off with attachment to some receptor. You get in througirgendwie generally it's clathrin-dependent endocytosis, so you wind up in an endosome, the early endosome, and then the late endosome, so the pH goes down as you progress. When you get to a low pH, say about 5.7, that triggers a conformational change in the envelope, which allows fusion and release of this nucleocapsid. I just want to go back once more to that concept of antibody-dependent enhancement, because that's shown here too. If you have a virus with, say, some antibodies on it, that one of those antibodies can bind to what's called the Fc receptor, a receptor present on some cells that binds to antibodies. And then you can go through the same process, although it's sort of indirect, receptor to antibody to virus instead of receptor to virus directly. So these are our class 2 fusion proteins. I don't know if you've done the classic influenza one yet, but so basically, so it's sitting on the cell surface in this dimer form. And then when you get to this low pH form, there's a major conformational shift where 3 of them actually form this ice cream cone-like trimer. And I think it's shown a little better on this next slide. So here are the dimers. And then at low pH, you get sort of rearrangement, and this little orange dot up here is a hydrophobic fusion loop that inserts into the membrane of the target cell. And then these things form these trimers, sort of rearranging to bring the 2 membranes together. This is all, driven by thermodynamics and, what's the most energy favorable state at a certain pH. So then you eventually bring the 2 membranes together where they fuse, and then you pull them apart, creating this little, essentially a pore. Remember, here's the viral envelope, so there's not actually a break in the membrane, but then the viral nucleocapsid can move into this part, which is the cytoplasm. It's just ribbon diagrams of the dimer and trimer again here. Okay. So the polyproteins, as I said, are a single RNA. They're all arranged for all these flaviviruses, or at least all the mosquito-borne ones, they're arranged the same way. They have structural proteins at the front end, capsid, PRM, envelope, and then this group of non-structural proteins, NS1, NS for non-structural, 1, 2A, 2B, 3, 4A. They were given these names after they solved the sequence of the genome. That's why they're in line. Previously, they had different names when they could just identify proteins. It is capped. Some of the HCV or the pestiviruses have an iris, but the genus Flavivirus has a cap similar to mRNAs, although it makes its own cap using a methyltransferase enzyme it encodes. There's no poly-A tail, but there are a fair amount of secondary structure in the back end here and some in the front end here. And there's actually a need for cyclization of this genome. So there's interactions between the back end and the front end that is important for the replication of the virus. And a lot of this, the interactions and the secondary structures, it's fairly crazy, and it makes my head hurt, so I'm not really going to go into that. But a lot of people have done a lot of work, looking at, how one specific nucleotide, if it screws up the base pairing of a stem loop back here, or an interaction between the 3' and 5' UTRs, can completely obliterate replication of the virus. So the protein, as I said, is translated more or less as one thing, and it's very highly associated with the membrane, so in and out of the cytoplasm. So here in the cytoplasm, you make the capsid protein, but then a signal sequence drives the rest of the translation into the ER, but then it comes out again, loops back in, comes out, loops back in. So all these things remain highly membrane associated. And inside the ER, host proteases cut at different spots. So then you release the capsid from the membrane, from the envelope protein, from the NS1 protein, which is actually ER luminal and secreted. And then the NS2B3 is a viral protease which makes cuts, on the cytoplasmic side, releasing NS2B3, 4B, 4A, NS5. So we know some of the functions. NS2B3 is this protease. It makes these cuts. NS5 is the RNA-dependent RNA polymerase. It copies the RNA into the negative and positive strands. It also has this methyltransferase activity, which makes that cap-like structure, as well as another methylation that helps protect the virus from innate immune functions in the cell. Oh yes, back to the dengue vaccine. Once you know this protein, the genome structure and protein architecture, people have made infectious clones of these things. So you have a cDNA clone, which you can use to make an RNA and put that in cells, and it'll make virus. And you could do standard molecular cloning things to mess with these things. So what one company called Sanofi Pasteur did was to take the yellow fever vaccine. So this is a pretty good vaccine. It's not pathogenic. But they took out the PRM and envelope protein. So these are the major targets of the antibody response. And they clone dengue 1 or 2 or 3 or 4 PRM and E into this yellow fever backbone. And then you can express that, and it made virus that is capable of replicating in cell culture. So then when you take all 4 of them, mix them together, and deliver them to a person, you can get an antibody response that then gives you some immunity against dengue virus. And they've gotten through phase 2 and 3 trials and actually are now approved for use in certain Central and South American countries. It's not actually a great vaccine, but they spend a lot of money on it, and they're going to sell that thing. And the other hope is that once you begin to produce more immunity, you cut down on mosquito transmission, and the whole thing gives you a positive effect in the end. And there's some other strategi' that are also in use in other vaccines, which are coming down the line in terms of clinical trials that involve just taking out parts of the 3' UTR, which attenuate the viruses. That was done at the NIH, and some other things done with some other attenuated forms of dengue virus. So I guess in the last, 2 minutes, 5 minutes, we're getting close to the end because the virus is getting close to getting out. Egress is still something of a mystery, but I included this because I think, just this visual slide is really cool. And this is done by electron micrographic tomography, which is taking an infected cell and doing EM slices across the cell. And then you can use computer algorithms to put the slices back together and actually kind of create these 3D structures of how the whole viral assembly process looks. I should say, as I said, the proteins are very highly associated with the membranes. And a Flavivirus infected cell has its intracellular membrane architecture goes kind of crazy. So you get all these big round vacuolar structures forming in the infected cell. And so let's just move to this middle one. What is proposed here, and there's a lot of other data in this paper doing antibody studies trying to show what's present in these circular compartments, is where replication is taking place. And what they propose is that virus replication takes place here, and then the capsid takes place in close apposition to the endoplasmic reticulum. And the capsid and RNA kind of move through this little juncture into the ER, and this little black dot here is a viral particle. So if you look at it this way, so the viral replicated in this yellow part here, and then pushed through the yellow part into the ER, and the emerging pink dot is the viral particle. It's maybe easiest to see in this particular slide here, where these are the replication compartments, where you have many of the non-structural proteins, the viral RNA, and then the capsid associates with the viral RNA, and it moves into the ER here. So then you get the envelope, which is present in the ER, so it essentially pushes into the membrane, creating the envelope membrane around itself, and then it moves through the secretory pathway, getting through the Golgi and trans-Golgi network, and it encounters furin and goes through that maturation process. So this protein PRM is the precursor of the membrane protein that's cleaved to a smaller form called M, and that's generally believed to sort of act as a chaperone to prevent premature fusion of these envelope proteins within the infected cell. Okay, so I've got about 2 more slides and 2 more minutes. So just hepatitis C virus is something I just wanted to quickly bring up, which was originally just described as non-A, non-B hepatitis. So it was some sort of hepatitis syndrome known to be associated with transfusion or blood product acquisition. That was not hepatitis A or B. In fact, hepatitis C has no relation to A, B, D, E. None of the hepatitis viruses are remotely related to either one. They're just all, it's the relationship between a banana and a toaster. They're all found in the kitchen, or in this case the liver, but that's it. Anyway, so generally it was believed to be mild, but it could be associated with chronic infection that could lead to much severe things, and often did: cirrhosis, liver failure, liver cancer. It was found to be a viral thing that could be passed to chimpanzees. We no longer use this research model, but it was fairly instrumental in sort of solving some of this. And it was cloned actually from entirely molecular means. Nobody was ever able to isolate it from an individual and grow it in cell culture. They were able to do it using a cDNA library and looking for things that reacted with HCV antibody. Still, since we were able to identify it and know the sequence, they were scientists were able to screen it out of the blood supply, so it's no longer a big danger for transfusion. Still, about 3% of the entire human population has chronic HCV infection, so it's still a fairly major problem. The architecture is pretty similar to what I just showed you, single positive sense RNA. It has 2 envelope proteins and a slightly different protein arrangement, but pretty much the same thing, same life cycle. No mosquito intermediate. This is bloodborne, right? So it is transmitted by, in mostly transfusion, transplant. Now its major source of transmission is shared needles in IV drug use. And the final point I want to talk about is just antiviral drugs. So people have been working on anti-HCV drugs for quite a while. Gilead had come out with an inhibitor of NS5B, that's the RNA-dependent RNA polymerase. Other groups have come out with inhibitors of the protease. So I won't dwell on this point, but these things are incredibly expensive, and yet they work very effectively. So as everybody runs out. The last point I just want to make is, so knowing the protein structure, we are able to develop antivirals. The cost is quite high, probably cheaper than a liver transplant and better in the long run. So you can weigh the value of that there. Yeah, so I'm out of time. And
thanks, Alec.
Yeah.